welcome back to another mini science lesson. Hopefully my mic works. Does it work? Is it working? Okay, yeah, the mic works. So today we are going to discuss genetic engineering, but before I do that, let me try and change my hat. There you go. Okay, so genetic engineering, what is it? Genetic engineering, it is, wait, let me write it. I'm trying to write here better. So genetic engineering, it's the direct modification of an organism's genome. Wait, no space there. I'll put it here. Direct modification of an organism's genome through the use of recombinant DNA technology. Okay, so if someone asks you, if a sensei or a senpai would ask you, what is genetic engineering? So it's the direct modification of an organism's genome through the use of recombinant DNA technology. And that will involve manipulating the genetic material of an organism, often by adding or deleting or altering the specific genes, so plus, minus, or just change that to achieve the desired traits or outcomes. And recombinant DNA technology would allow scientists to combine the DNA from different sources, and that could create genetic combinations that would not naturally occur, and that's commonly used in agriculture to develop genetically modified organisms. Wait, let me just grab the water. Grabbing H2O. One moment. Alright, I'm back. So, what are the key aspects of genetic engineering? Let's see. For that one, you can do cloning. Wait, let's erase. That is direct manipulation of a genome using recombinant DNA technology. Alright, so key aspects could be gene cloning, aka you isolate and copy a gene of interest. You can do gene editing when you just modify it using like CRISPR, Cas9. And you also have recombinant DNA, where you combine DNA from the different organisms to create new sequences. Gene therapy is also possible, where you correct the defective genes in a patient's DNA to treat the genetic disorders. The gene therapy, again, it's the correcting defective genes in a patient's DNA. Just gene cloning is just you clone genes, you copy, isolate and copy a gene of interest. Gene editing is when you edit a specific sequence. Recombinant DNA is when you get DNA from different organisms and combine them. Gene therapy is when you try to fix a patient's gene to treat genetic disorders. And then you have GMOs, the genetically modified organisms like crops or animals, to exhibit the desired traits such as increased yield or pest resistance or disease resistance. Okay, and so what do we need genetic engineering for? Well, you need it for lots of things. It's important for both research and commerce. What is that hat? I will change that in a bit. Alright, so Genetic engineering is useful for research and commerce. COMMERCE. Because in research it is used to investigate biological phenomena, and that would allow the scientists to explore and manipulate the genetic makeup of different organisms to better understand their function, behavior, and response. In commerce, you can produce the different drugs and food for it. So yes, examples would be Boyer and Cohen patents for recombinant DNA, and that has actually approximately gave Stanford University about 250 million USD in royalties. So that's a lot. And then Columbia University received approximately 800 million for their DNA co-transformation, and that patent covers the method of transferring DNA into eukaryotic cell. So see, genetic engineering really plays a critical role in advancing scientific discovery and commerce industries. But why is it hard? Why is genetic engineering so hard? Because you can just imagine, oh, I'll just cut it and then I'll just change it. But what makes it difficult? Well, what makes it difficult is, first, this hat I don't like the octopus hat. Okay, there. So what makes genetic engineering difficult is organisms have four things. One is protection mechanisms. Organisms would has evolved various defense mechanisms to actually protect their genomes, so you can't just change it willy-nilly. And that includes immune responses and apoptosis, DNA repair mechanisms, and all that. We discussed it before. And then on a molecular scale, it's very tiny, right? It's very tiny. So, and it's difficult to directly manipulate it because of the small size of the different components. And it's very complex too. There's also the part where the efficiency is low. And even if you try many attempts, maybe only a small fraction of the changes or edits may successfully occur. And lastly, you can only the success is only indirect success determination. And that's because even when you have a desired trait or change made, it might take additional experiments to fully understand the change that you tried. So yes, why is genetic engineering hard? It's because of the protection mechanisms. It is on a molecular scale, low efficiency, and it's just indirect success checking. Okay, so let me just put a sticky note here. Sticky note. Okay, so what is the perfect genetic engineering strategy? Time to erase it. Where is the pencil? Oh no. Oh, I was using the other side. Here you go. Welcome back, Elmer. Thank you for coming back to the stream. Oh, since you chatted, your character will pop up soon. Mini character at the bottom. There you go. It's there. And you can change your hat by typing !hat. Okay, so what is the perfect genetic engineering strategy? Well, first you target it or not. Take this bit, and that would be with a specific location in the genome, or introduce it randomly into the genome. The targeting provides more control, over where the modifications are made. And then you also have homologous versus random integration. So with homologous integration, you just insert the genetic material into a specific site on the chromosome, and that would match the inserted sequence. Random integration would allow the inserted DNA to integrate at random locations. And then we have the positive selection and negative selection. So positive selection is only the cells that have successfully integrated your desired genetic material will survive or thrive, and that would be achieved if you introduce a specific marker, a selectable marker. Negative selection is when it will ensure that cells have taken up the unwanted genetic modification. So the ones that took the negative one or the unwanted one are guaranteed to be eliminated, or at least reduce the likelihood of the unwanted effect. So that's why it's positive or negative. Spatial control is the fifth one. Spatial and temporal is the sixth. Spatial is that's the ability to control where the introduced gene is expressed. Temporal control involves the timing of when the gene is expressed. Okay, so that's the perfect, quote unquote, perfect genetic engineering strategy. So one is targeting, and next is, water break. Okay, so that is the perfect genetic engineering strategy. One is you target homologous or random integration. There's positive and negative selection, and there's spatial and temporal one. Positive selection is when those cells that took up the genes that you like will be the ones that will survive or thrive, and then negative selection is the cells that took up the genes that you don't like will be eliminated. And then spatial control is just that's the ability to control where the gene is introduced. Meanwhile, temporal is when the gene is introduced, the time. And all of these will influence all of those would influence the precision and effectiveness of and effectiveness of the genetic engineering projects that you're trying to do. Okay, so what are the different techniques that we should know? Well, let me write them down. Color. There you go. So the different techniques that we should know are, first, blue pen. Okay, number one is knockout. Genetic engineering techniques. One is knockout, knock in, and knock down. Okay, what do these mean? So knockout is when a gene is completely removed or inactivated. Knock in is when a specific gene or mutation is inserted into a particular site. Knock down is when the gene expression is reduced, often by using RNA interference. So one more time, knockout was when the gene is completely removed. Knock-in is when the specific gene or mutation is inserted into a particular site in the genome. Knockdown is when the gene expression is reduced. Okay, number 2 is insertional mutagenesis. What is this? It is when a foreign gene is inserted into the genome and that could disrupt the function of the other ones. So insertional mutagenesis, you insert a foreign genome and then you disrupt the function of the other genes. 3 is transposon based mutagenesis. And that would use transposons or DNA elements that can move within the genome to introduce the mutations and study the gene function. So what is transposon-based mutagenesis? That is using transposons or DNA that can just move freely in the genome to introduce mutations. And then you have the 4th one, which are viral vectors. So viral vectors, that means viruses are used to deliver genetic material into cells, and that would be commonly used in gene therapy or transfection experiments. So viral vectors would mean using viruses to deliver the specific genes that you want. Then you have chromosome engineering. It is a technique where you manipulate the structure or function of the chromosomes. This is a chromosome. And an example would be a chromosome deletion or rearrangement. Next, you have gene trap mutagenesis. And that is a method that inactivates genes by inserting a reporter gene into them. And that would help identify gene functions based on the reporter's expression. Okay, so what are the differences? So far, knockout means you completely remove or deactivate a gene. And then knock-in is when you try to a specific gene or mutation is inserted. Knockdown is the gene expression is just lowered. And then you have insertional mutagenesis, where you insert a foreign gene and then the foreign gene will alter, you cause the mutations. Transposon based mutagenesis is when you use transposons, which are DNA fragments that can freely move inside the genome. And those will introduce the mutations. Viral vectors is when you use a virus to deliver the mutation. Chromosome engineering is when you do deletion or add on of a part of a chromosome. Hello, George Roas. Yes. Wave emote. Thank you. Welcome back. We are discussing genetic engineering and some techniques for it. And I was trying to mention how viral vectors is when you use a virus to introduce a mutation into a gene. Chromosome engineering is when you manipulate the structure or function of chromosomes, such as chromosome deletion or rearrangement. Hello, Lendor. Yes, hat. Whoop whoop. You immediately changed your hat. Nice, you got the emote. Yeah, you can feel free to keep trying. I think there's more than 50 hats at some point over there. So yeah, have fun. Number 6. We are now at number 7. Number 7. This is the BACs, PACs, and YACs. Okay, what are these? So one is bacterial artificial chromosome. PAC is P1 artificial chromosome, and YAC is yeast artificial chromosome. What are those? They are just used to clone large fragments of DNA in the bacteria. So artificial chromosome, essentially. One moment. Water. It's the air is so dry. Okay, next. Recombinering. Wait, I have to write it. Number 8. Recombinering. There you go. So that one is a method of genetic engineering using homologous recombination, okay? That one is you insert or modify genes in a bacterial or mammalian cell. So again, a method of genetic engineering using homologous chromosomes. Recombination, just recombination. And then the next one's 9 to 13. They're just very specific techniques. Since we are out of space, let's make space. Here. Alright, there it is. So number 9 is Krelox or Krelox and Flip FLPFRT. What are that? What are those? Those are recombinase systems just to specifically recombine DNA at defined sites. So see, they're just examples. There's also conditional gene activation or inactivation, where you just inactivate or activate genes in a controlled manner, usually in a response to an external signal that you put. Conditional gene activation again is when you just trigger a gene on or off depending on an external signal that you put. And then you have conditional gene, you have zinc finger nucleases. Oh, by the way, I think I mentioned in a mini announcement that we are going to have, what's that called? We're gonna have a music video that finally has music in May and one in June as well. Whoop whoop. So watch out for that one. Any guesses on when it will be released? That's too much of a spoiler, I think. So yeah. And then now we have CRISPR, the most famous one. I think. Or except for the knock and knockout. Yes, dab. Yeah. CRISPR is... Oh, not... I knew you were dabbing for the music video. Yes. And then CRISPR, this one, it has each letter means something. Okay? I think I have to know this. So CRISPR, clustered, regularly, interspaced, short, palindromic, repeats. What does that mean? Okay, calm down. I'm telling myself to calm down. It's okay. Clustered, regularly interspaced, short palindromic repeats. Clustered, regularly interspaced, short palindromic repeats. Clustered, regularly interspaced, short palindromic repeats. What does that mean? Just so we don't have to brute force memorize it. So, if you have palindrome CRISPR, yes, aside from the mitochondria as the powerhouse of the cell, this is something that should be stuck in your dreams. Clustered, regularly interspaced, short palindromic repeats. So short palindromic repeats, what is a palindrome? Let's start with that. You know race car? If you spell it backwards, it's still race car. So that is a palindrome, a word that you can spell backwards and it's still the same thing. So now, it says short palindromic repeats. Let's start with that. So if you have race car, that is the palindrome. If you mean short palindromic repeats, you just have race car. And then it is clustered but regularly interspaced. So the race cars are just regularly interspaced. Why is that a hard word to say? Regularly interspaced. Clustered, regularly interspaced, short palindromic repeats. One more time. CRISPR is clustered, regularly interspersed, short palindromic repeats. Okay, we got it. It's okay. Now erase the rest because there's still the 13s. 13. Clustered, regularly interspersed, short palindromic repeats. Sticky note. I have to remember that one. Sticky note. That one is when... So CRISPR, actually, is just a revolutionary genome editing technology where you use a guide RNA to direct the Cas9 nuclease to a specific DNA sequence. That is DNA. Look at that. That's art. Fine. I'll try to write it the squiggly properly. See, there you go. So again, that is just where you use a guide RNA to guide the Cas9 nuclease to a specific DNA sequence that you want. So it's just it allows precise editing of the genome. Alright. And then you have the TET or LAC. What is that? So for the TET, that is just a system. Oh, I did not write the 13th or 14th. 13, that is TET, LAC, and GAL4/UAS. These sound like the idol. I don't know. It's like TXT, BTS, EXO. It's like that. TET, LAC, and GAL4. Okay. So TET is the tetracycline system. It's used for controlled expression of genes in response to tetracycline or doxycycline. And then the LAC system is just, it involves the regulation of gene expression by the LAC operon. You will always hear about that in your biology classes. GAL4 or UAS is used in genes and other organisms to activate or repress the gene expression using the GAL4 protein. Yes, that's all, those are so far the strategies that I mentioned. Again, just writing them all out again. I'll try to make them fit in one screen. So we have knock in, knock out, knock down, knock down. So knock in is when you introduce a mutation, or introduce a specific gene or variant in it. Knock out is when you remove or inactivate a specific gene. Knock down is when its gene expression is just lessened. You have insertional mutagenesis, where if you insert a foreign gene, genesis, then it causes some mutations to occur. And then you have transposon-based mutagenesis. That is when you use transposons, aka DNA sequences that can freely float around in the genome, and those would introduce the mutations. 4 is viral vectors, where you use viruses to introduce the variant. 5 is chromosome engineering. That is when you just edit a specific part of the chromosome. 7 is the BACs, PACs, and YACs. That is the bacterial artificial chromosome, P1 artificial chromosome, and yeast artificial chromosome, just to clone large fragments of DNA. Number 7 is, that's 7. What was 6? This is 7. Number 6 is gene trap mutagenesis. It is a method that inactivates genes by inserting a reporter gene. 8 is recombinering, just another term for genetic engineering using homologous recombination. Crelox is just you use a recombinase system to recombine DNA at specific sites. And then you have conditional gene, 7, 8, 9, 10. You have conditional gene inactivation, or just activation, where you turn them on or off using an external signal. 11 is the zinc finger nuclease, where engineered proteins will be used to create the double-stranded breaks, or DSB. Number 12 is CRISPR. What is it? Clustered regularly interspaced short palindromic repeats. That is where you use a guide RNA to guide the Cas9 nuclease to a specific part of your genome, or the specific DNA sequence that you want. And 13 is just the TET, LAC, and GAL4 tetracycline, lac operon, and then gal for protein. Whoops. Next page. Okay, now how do you recombine large plasmids? That's a great question. And first I will have to sticky note this and then erase. Erase. We wrote a lot. Okay, so here, when you want to recombine large plasmids, that would require working with long DNA fragments that can't be manipulated easily with classic cloning methods. What does that mean? Well, that's because you gotta use like homologous recombination, HR, to engineer the targeting vectors, and then you gotta use Rec-A from the Rec-A protein from E. coli, and that will be used for recombination as well. So what are the steps? There's 4 steps. 1, 2, 3, 4 if you want to make large plasmids. One is shuttle vector creation. Okay, so the shuttle vector will be constructed, and let's say that's a ship, and you can have the different components that you need, like the lac-Z, the tet, the rec-A elements, and the origin region for replication. After that, homologous recombination happens. Okay, and the vector will recombine at either the red or blue regions using the rec-A. So first is you make the shuttle vector, which has all of the things that you need, and then you have homologous recombination, which recombines at either the blue or red regions. And then you have the selection process, and at that point, selection process. And at that point, you expose your vector to tetracycline for selection at around 43 degrees Celsius. And then further recombination happens if required. So essentially, you have the shuttle, which carries all the things you need, homologous recombination, and the selection process. And then if we want, next is, what about the viral vectors for gene transfer? Viral vectors. For that one, it is well studied in biology, right? So viral vectors are very widely used because they are well understood, and that makes them reliable tools for gene transfer. And the vectors would usually, they kind of look like this. Sometimes in a circle and another circle. Yep, that's a gene. That's a viral vector. So the vectors have a cloning capacity of around 10 kilobytes, KB, allowing them to carry genes of a certain size into the host cell. Okay? And that will they will deliver in 2 ways. One is they can either carry the gene in an episomal form or integrate the gene permanently. So an example is adenoviruses would just do transient gene. Their adenovirus would be transient. Meanwhile, retroviruses would be more permanent. So that's why you kind of want to use retrovirus if it's for gene therapy and research. But it also depends on the use that you want. So adenovirus is more for transient, meanwhile retrovirus is more permanent. Okay, next, gene traps. I mentioned it a little bit before. What are gene traps? Traps everywhere. Gene traps are just used to identify unknown genes or regulatory elements like the promoters or enhancers within a genome. And the trap works by gene trap. The trap works by inserting a random or inserting a reporter gene. Here, insert a reporter gene with control elements. And the inserted gene enables the identification of various gene regions or regulatory sequences in a model organism's genome. And another type of gene trap is when you detect genes based on the spliceosome site located near the promoter. So yeah, essentially just trying to find unknown genes. There's also the promoter trap to detect the presence of gene promoters. Just to detect the trap for a gene, to find unknown genes. That's what the gene trap is. And then we have the knock-in, knock-out. Okay, how does that work? The process of knockout and knock-in is a very fundamental technique in genetic engineering, okay? Let me erase the gene trap info. In genetic engineering, knock out, knock in. So here, that will be you modify genes in organisms, especially in mice. And that in gene targeting, a gene interest or X is targeted for modification. So let's say you have this is gene X, and you want to change this part. Then you have a construct, this is this one, this, and you want to put this part in there. Okay, so the construct is introduced into a cell which has the gene for selection, and then you have the virus, like a herpes simplex virus, that allow for negative selection. This will integrate the genome using homologous recombination, HR, and that will replace the gene X with the one that you like. So the final output is this. Yes, now you have the red part here also. And then you have in random integration, it's just everywhere else. And that method is essential to create the gene knockout or knock-in. So what are the steps for it? Steps for knockout or knock-in would be, well, just focus on mice. Let's erase it first. Focus on mice. Yes, hat change. Okay, knockout technology involves crucial steps. So right now we are in knockout. Oh, it reminds me of Tekken. KO. No, stop. Science first. Tekken later. Knockout is first is integration of the targeting vector. First step is you integrate the targeting vector inside the gene of interest. And that is through homologous recombination in the embryonic stem cells. We have this. Draw it out. Here. This is your embryonic stem cells, right? Or iPSCs, for example. Hello cheeseburger. Hand wave. I will use the hand emote. There you go. There, because we have a wave emote. Yes. So embryonic stem cells. After that one, I cannot read the image, it's too small. Anyway, just integrate the targeting vector inside the gene of interest through HR in your embryonic stem cells. And then you have positive negative selection to isolate the cells that you have. So imagine now you have the cells that you have. Yes, I mean that you like. That's a star. That is a star. Star. I can do a star. So now you try and put that inside mouse blastocysts. Number 2 is injection into blastocyst. And that is where the blastocyst, after that, the blastocyst is transferred to a pseudo-pregnant mouse. To a mouse. This is a mouse. 1, 2, 3, 4 legs, I think. And the ears. So, okay, again, you integrate the gene of interest into your decent enough star. Thank you. Oh, the tiny head of a star. We can make it. There you go. Okay, now its left leg is too small. It's alright. A star is a star. So you have a targeting vector. You use your vector to deliver the gene of interest using HR. And then after that, you isolate the ones that you already have. Then you put it inside a blastocyst. Once you have a blastocyst that has your gene, you put it inside a mouse, a pseudo-pregnant mouse. And then the mouse could exhibit they will give birth to a litter of chimeric mice, which could be a mix of modified and non-modified mice. So NM. And then you just distinguish them using different coat colors. After that, after that one, you will breed the chimeric mice with non-transgenic mice. That means the mice that you did not edit. And so the resulting progeny or the resulting offspring, you will genotype those and to identify the transgenic mice. Note that only some might have the thing that you want, so you have to check if you get the homozygous and heterozygotes. Check those, because you might have, you just have to make sure that you're using the right mouse. Okay. Crelogs, nah, that's fine. DNA crossover by site-specific recombinases, just crossover, bro. And then estrogen receptor is a system used for conditional recombination in genetic engineering. And then now we have CRISPR. CRISPR-Cas. What is CRISPR? What is the clustered regularly interspaced short palindromic repeats? Good. CRISPR/Cas, it is a technology used to target used for targeted genome editing. It's very precise. And then you use a guide RNA to deliver the Cas. It will just guide. Now you can't remember that one. I have to type it. Clustered regularly interspaced short palindromic I cannot spell. Letter P. Palindromic repeats. Nice. That will be, it will be funny if I pin it. It is now pinned. Clustered regularly interspersed short palindromic repeats. So, how does this work? Okay, we have to write the steps. Actually, CRISPR, it forms the basis. Wait. It forms the basis of an adaptive immune system in prokaryotes, and like bacteria and archaea. So it's also important. The process begins when an organism, it's like story time. Once upon a time, CRISPR happened. Custard regularly interspaced short palindromic repeats. Okay. The story begins when an organism acquires fragments of foreign DNA. So it gets foreign DNA, such as plasmid or viral DNA, and you insert that into a CRISPR loci in the bacterial genome. I'm gonna use purple, violet. Is this purple or violet? It's halfway, both of them. This is, you put the foreign, like a plasmid DNA into the bacterial genome. Okay, and then you transcribe the CRISPR loci into the precursor, and that is processed into smaller cRNAs, which guide the Cas9 proteins to the foreign DNA. The Cas proteins to the foreign DNA. And the Cas9 would then cleave the target DNA at that sequence. And so it's very, very exact. So again, how do we have the exact steps for this one? No. It's okay. But yeah, that is the concept of it. You have your plasmid DNA that contains the thing that you want, and then you put it inside a bacterial genome, and then it becomes a lot of cRNAs. And then after that one, it will guide the Cas proteins to the DNA region, and then the Cas9 will cleave the target DNA, providing at the corresponding sequence, and that will provide lasting immunity against subsequent infections from the same DNA. And in CRISPR, what are the three components of clustered regularly interspaced short palindromic repeats? The three components are, number one, that is the TRAC RNA. It's very common. Two is cRNA. It's unique to the target. And then you have the Cas nuclease. So you have the TRAC RNA, which binds to the cRNA, forming a hybrid that interacts with the Cas9 protein. Cas9 would target and cleave the foreign DNA at the site. See, they all talk. Communication is key. What? They all talk to each other. See, wait till you see the signaling. Oh my gosh, I didn't talk about that yet. Soon, it's the GPCR, the map, all of it is connected. Okay. Next is double-stranded DNA breaks. Yeah. That's typically introduced by Cas9, and that would lead to a lot of mutation types, and then the non-homologous end joining would just result in insertions or deletions. Okay, so why is Cas9 important? That's because it provides a broad range of genetic engineering possibilities, which I will write. Cas9 here. CRISPR Cas9. Clustered regularly interspaced short palindromic repeats. Nice. It reminds me of pneumono ultramicroscopic silica volcanoconiosis. That it's like that, but it's just a tad bit shorter. Indels, insertions, large. Okay, yeah, this is the important stuff that Cas9 gives us. It gives us indels, which is a Cas9 would induce the double-strand breaks, and then it would cause the repair mechanism to create insertions or deletions. Indels. And then you have insertions or replacements. And then you have large deletions, or rearrangements. And then you have gene activation. You have chromatin or DNA modification. And then you have fluorescent protein labeling. So see, CRISPR-Cas9 gives a lot of things. Insertions or deletions, insertions, replacements, large deletions, gene activation, chromatin or DNA modification, or just fluorescent protein labeling. It's just a mini review. What is genetic engineering and how does recombinant DNA technology play a role in modifying genomes? So we know genetic engineering is the directed modification of an organism's genome using recombinant DNA technology. And a recombinant DNA technology allows for manipulation of DNA by combining the materials from different sources. So again, genetic engineering just provides, whenever I memorize it, it'll be like, oh my gosh, P. Sherman 42 Wallaby Way, Sydney, yes. Oh, Finding Nemo was really nice. I the fish part where they were inside the, okay, everything was a fish part, but it was when they were trying to save Nemo inside the dentist, that part, it was the dentist scene. I kept laughing. And then Finding Dory was also another story. Here we next. So yeah, what is genetic engineering? It's just the direct modification of an organism's genome using DNA recombination. Okay, explain the difference between gene cloning, gene editing, recombinant DNA, and gene therapy. Gene cloning is just when you make a copy, isolate and copy the gene to get another one. Gene editing is when you just cut it up and edit directly using Cas9. Recombinant DNA is when you use recombinase technology or combine it from different organisms. And gene therapy is just gene therapy, I know it's for patients and you correct the defective genes in patients to target genetic disorders. They describe the role of positive and negative selection. That is only the cells or organisms in positive selection. Positive selection. Positive selection, only cells or organisms that successfully incorporated your gene of interest will survive. And then if you have the negative selection, the cells with undesired modifications are eliminated. Okay? If they did not incorporate the construct that you used when exposed to specific drugs, eliminate them. How does homologous integration differ from random integration? Homologous is when you insert it at a specific location, while random integration is literally just random. So it's less controlled. Meanwhile, why is genetic engineering difficult? And that's because there were 5, I think. One, there was like low efficiency. Your organisms would have their own protective mechanisms, and then you can only get the result indirectly, so there is no way to get it. Oh my gosh, yes, this is exactly like Pal World. Thank you for making that connection, because I was trying to make a legend divine dragon musclehead burly body for a gazillion jet dragons. They're not happening yet. I think I got one, but I need a legend divine dragon, infinite stamina, and a musclehead. No, runner. So I have a speed dragon that's also kind of strong. Soon. But so far I was able to build one. What was it? The advanced incubator that needs a gazillion hexalites. I was able to make one. Finally. Anyway, so that is genetic engineering and Pal World. Oh my god, I changed the title of this one to include Pal World and breeding. That is funny. Yeah, eliminate the ones that you don't like. Just condense them. I don't butcher, I just condense. Yeah, what is the purpose of creating genetically modified organisms? The purpose is for so that you can defeat science. I'm kidding. So the purpose is you want to introduce specific traits that improve the organism's functionality, such as pest resistance, increased yield, or disease resistance in crops. Hello. Thank you for the many trout. Yes. We are discussing genetically modified organisms and connecting science with Palworld because of course everything is about Palworld or Pokemon. Nice burger. Okay, so we want to create genetically modified organisms, aka good jet dragons and good Jormundside ignises, to improve the organism's functionality, such as pest resistance, increased yield, or disease resistance in crops, or in the case of jet dragon, so that it is stronger. I don't know if it's gonna be super tanky, but if it has burly body, then there's plus 20% defense. Haha. Okay, now explain the concept of gene knockouts, knock-in and knockdowns. Knock-out, we know this. Knock-out, remove the gene or inactivate it. Knock-in, put a gene that you like into a targeted site. So if you have the runner passive solo in one jet and you want to put that in the other one, that could be knock-in, maybe, if you think about the passives as the genes or genetic modification that you want. And the knockdown is the expression of a gene is reduced. Discuss the differences between CRISPR and the others. CRISPR, again, clustered regularly interspaced short palindromic repeats. Nice. It is a gene editing system that uses the RNA, guide RNA, to direct Cas9 to a specific DNA sequence. It will cleave it, and then if you want to know, it is easy to design, it's highly precise, unlike breeding the specific mouse that you want. It's not very precise, you just have to hope that the RNG happens. And the limitation of this one is potential off-target effects. You might get ugly passives, sadly. So, if we want to design an experiment using CRISPR-Cas9 to knock out a gene of interest in a model organism, how would we do that? So let's say we're selecting a gene of interest like the TP53 in mice. You design a guide RNA first. You design a guide RNA complementary to the gene of interest. I think I have to write that just to make sure, because I'm a visual person. I think anything that can help us make this stick into our brains. So we have, you did first, so you have a gene, this is your gene of interest. I mean, that is this is the target region, okay, target region. And then we design a gene, we have a specific gene of interest, TP53. You design a guide RNA, which is complementary to that one, and you introduce the CRISPR-Cas9 into the mouse embryos via microinjection. And then use the PCR and sequencing to confirm if the gene was knocked out. The expected outcome is the mouse singular, mice plural. So the mice with a deletion in TP53 would have tumor susceptibility, because TP53 is important in tumor suppression. And of course, if you have deletion on that one, then you're then you're screwed. Not you, the mice is screwed. Hopefully you don't have a deletion in that one, please no. Okay, how would you use a viral vector to deliver a gene into a target cell? Well, you choose it first. Retrovirus for stable, adeno for transient. Adeno is temporary, retro forever. Retro has a lot of R, just like forever. Okay, you choose the viral vector. I'm going to choose retro for the forever part. You package the gene of interest in your vector, infect the target cells, and then you select the target cells using a marker gene. Okay, so again, you choose a viral vector, you package your gene inside that, you infect the target, you infect your cells with it, and then you check using a marker gene. Adenoviruses again are what? Transient, not permanent, temporary. And then retrovirus is more permanent. If I want to create a transgenic mouse with a gene knockout, how should I do that? Well, I will design a target vector with my marker. You flank it with homologous arms, and then you use electroporation to introduce the vector in your embryonic stem cells. Design, we talked about this one. Okay. Finished. Oh, if I want to design an experiment to introduce large deletions into a genome using CRISPR-Cas9, what controls would you include and what type of outcomes would you expect? So we can design 2 guide RNAs to flank the region to be deleted. And then you co-inject these with Cas9 into mouse embryos. And then you perform PCR. Finished. If we want to create a model organism that expresses a gene at a specific time, remember temporal, how can we do that? Well, if you want to use the tetracycline on system, you have a tetracycline responsive promoter which drives gene expression. If you introduce the gene for a tetracycline controlled transactivator and place it at the gene of interest, then if you administer doxycycline, you can activate gene expression at a specific time. Yes. And then, so far transgenesis, we finished it. Next is experimental design in a transcription. Whoop whoop. Okay, now calm down. It's okay. It's gonna be fun. I'm telling you that much. Yeah, it's okay. So, what is experimental design? You design an experiment. Oh, bye. Thank you for coming to the stream. Oh, the burger waved. Yes, thanks, Landor. Okay, to understand the molecular mechanism behind the change in biological activity, specifically the increase in sodium, well, you can break it down into different steps. You can activate them by phosphorylation or other modifications. There is also protein content changes. I forgot, I have a whiteboard. I gotta use this. Here. There you go, I'll type it. No, right. Number 1 is activation by phosphorylation. And then you have protein content changes. Okay, if the thyroid hormone leads to an increase in sodium, potassium ATPase through the protein changes, then it's most likely post-transcription or transcriptional regulation. Voila. Okay, so protein content, that is the one we will check, and the main question is it transcriptional or post-transcriptional? We have a mini map that we gotta remember, aside from the clustered regularly interspaced short palindromic repeats. Okay, so this is transcriptional and post-transcriptional. Transcriptional control of protein content. Okay, before I continue, tubing break, the water. Okay, got him. Okay, let us start from DNA. DNA. Start with DNA. Then you have RNA, right? You have the RNA. What is that arrow? I think can I make it smaller just to make it fit? 5. Okay, you have DNA. Yes, water. We are water, earth, fire, air. Long ago, there were 4 nations lived together in harmony. Then, everything changed when the Fire Nation attacked. Only the Avatar, master of all 4 elements. Master of all 4 elements could stop them. But when the world needed him most, he vanished. 100 years passed, and my brother and I discovered the new Avatar, an airbender named Aang. And although his airbending skills were great, he has a lot to learn before he's ready to save anyone. But I believe Aang can save the world. Yeah, it's echoing. Okay, DNA. Then you make the RNA transcript. This is where the first control happens. Nickelodeon and Disney before. Awesome. I still remember the Danny Phantom. The not music video. The opening. Wait, this first from DNA to RNA transcript. This is the first one. This is transcriptional RNA. No, transcriptional control, normal. And then RNA transcript to mRNA, that is RNA processing control. Because this is where, remember in the previous video, this is where the 5' capping happens. 5' capping, splicing, and poly A tail. Yes. And then after that, it goes from the nucleus. It goes out actually. Nucleus. This is a nope, not green. Green is for mitochondria. Green is for mitochondria. So I don't want to mix that up. This is the nucleus. I'm gonna make this a tad bit bigger. This is the nucleus, and then this is the whole cell. The whole cell outside. Here, this is the cell. It's a big cell. It will go out from the 5px. This 4, it's okay. So from mRNA, the mRNA will go out. And what kind of control is here? RNA transport. And then from mRNA, you have, it could be used, it could be not used. You can make it into inactive mRNA. That is where, we are now in the cytosol. That is where mRNA degradation control happens. That's number 5. I know, I skipped 4. Just wait. This is 5. Oops, no. Here, 5. This is mRNA degradation. Yep. And then, if it is used from mRNA to what? What is it made into? A protein. So here, this is translational control. Number 4. Translational control. After protein, you can have an inactive protein, and active protein, the one you use. And what is here? That is the protein activity control. So number 6. See, there's a lot. 6 so far. So yes, transcriptional control regulates the process by which genes are transcribed into RNA. And then that is influenced by specific transcription factors and DNA elements. And then you have RNA processing, which includes splicing, capping, and poly A tail, preparing the mRNA for translation, and then the process mRNA is then controlled during RNA transport, where it's moving from the where nucleus to outside to the cytoplasm. And then once it is in the cytoplasm, translational control happens, whether and how much protein will be synthesized. And if you don't like that, you just have mRNA degradation breakdown to prevent the production of unwanted proteins. Finally, you have protein activity control, which takes place after the protein is made or synthesized, and with post-translational modification like phosphorylation or acetylation, determining whether the protein is active or inactive. And then we have techniques. Hurrah! We have different techniques. Hello Kaizeki, welcome to the stream. This is a very nice scribble of transcriptional and post-transcriptional control. And by the way, you have a character now because you chatted yes. It's automatic and you can change your hat using exclamation hat. So that's why you're seeing exclamation hat there sometimes in the chat, because you can change your hat. There's like 50 hats or something. I lost track. Yeah, just have fun there. So for protein and mRNA quantification. Why is my handwriting flying? It's okay, you know, it reflects your mental stability. Quantification. You have protein, which you can use Western blotting. No, it's okay. You can just listen as well. But it is science. Life is science. Science is life. And this is technically also genetic engineering connected to Palworld, because we were talking about traits earlier and trying to put legend, divine dragon, musclehead, and what was it? Burly body, and that is just making the perfect attack Ragon. Actually, I can put ferocious here if it's just a glass dragon that's just super strong, but then it'll be immediately killed. So I just like putting burly body so its defense is also increased, so it won't die immediately. So yeah, for mRNA, you can use northern blotting, qRT-PCR, and then microchip analysis, and RNA-seq. So what are these? So for protein quantification, you can use Western blotting, which will detect and quantify specific proteins within a complex mixture based on their size and antibody recognition. Meanwhile, for mRNA quantification, there are advanced techniques. Northern blotting is less common due to newer methods, and that will allow the analyzing gene expression and RNA processing events. And then you have qRT-PCR, quantitative reverse transcription polymerase chain reaction. Ooh, hat. I want to try and find the dab hat. It's okay. Someone has the dab already. That's awesome. I have the haha hat. So qRT-PCR, it's a very modern and highly sensitive method that quantifies gene expression by amplifying specific mRNA sequences, making it particularly valuable for measuring low abundance transcripts. I mentioned microchip analysis here because microchip analysis will use DNA microarrays, where thousands of gene probes are placed on a small chip to do gene expression. RNA-seq is it offers a way, it's a comprehensive method for transcriptome analysis. It sequences the RNA molecules, providing an in-depth profile of gene expression. Right, so if someone asks you, how do you do a Western blot? I have to answer it. Okay, so let's try. Let's do the steps. One moment. Western blot, Western blot. Erase these first. Erase it. This is the moment where we draw Western blot. Western blotting. So first step, number 1. Load and separate the proteins using SDS page. What does that mean? One moment. Load and separate protein samples using SDS page. So SDS page, that is a specific type of gel electrophoresis. It is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Again, sodium dodecyl sulfate polyacrylamide gel electrophoresis. Proteins will be separated based on their size. How? Okay, we have this kind. It looks like this. You have, oh, it looks like a castle. Sakurajima castle. I want to build my Sakurajima castle soon in Palworld, after we do Western blotting. So you load it, and then they will travel from up to down, or depending on the direction. But yes, after the proteins are separated, they are transferred from the gel to a membrane. Gel to a membrane. I will just write here. Gel to a membrane. Whoops. Gel to a membrane. Where is it? Protein to immobilize. Yeah. It is a PVDF. Oof. PVDF. Polyvinylidene fluoride membrane. And this will allow the protein to be immobilized on the membrane to make it stuck there. Here. The gel, this is the membrane. You put it all in. This is the negative side, this is the positive side. You have the gel here, and then you have the membrane at this. This is the second membrane. Perfect. After that, the membrane, this is step 2. After that, step 3, you block the membrane with neutral protein. Okay, this is the membrane now. I'll make blue. Blue. You block the membrane using neutral protein. Why? That will prevent non-specific binding. So only the proteins that you like will, like later on. This is important. This is an important step. Okay? To prevent non-specific binding later. After blocking, the membrane is incubated with, primary antibody specific to the target protein. So take note, so far, your proteins were able to like travel, nope, not the red, I want red. Travel down, right? Let's say you have this protein over here. It is now there, and now it's over here. This is the protein. When you block the membrane, the rest of the early proteins that you like will be blocked, and only this protein will be available now. And then, after you block, it says to incubate it with a primary antibody. I will use orange. Primary antibody. One primary, AB. That one will attach to your protein. So you have this. Is your protein. I will write it. And then your primary antibody will stick to it. Yeah, like that. And then after that, so now you have selective binding, right? And then you have your secondary antibody next. And we'll just write it in 5. Secondary antibody will attach to it. So the next picture will be, you have the red one. You have the orange one. And then your secondary AB will be here. Yes, and that one will emit light because the secondary antibody helps amplify the signal for detection. And then you, just lastly, you will, number 6, incubate it with an HRP substrate. Take note your secondary antibody will be, so far for this one, it's an HRP labeled, right? But the substrate will then help like emit light because your HRP substrate reacts with the HRP here, and then you get sparkly kira light, and so you can see the band, the intensity of the band that will correlate with the amount of target protein present in the sample. Woo! We did it! We did Western blotting. This picture looks so pretty. Look, I'm gonna move over here just so you can see. Yeah. Okay, before I continue with Northern blotting, water. Yeah. Okay, I drank water. No. Feels like when I was in AP freshman science where my teacher showed so much papers on my face. Don't worry, no papers will be showed today. This is just a drawing. Now let's do Northern blotting. Wait, when you mean AP science? Oh, okay. So that will have the biochem and physics also. Cool. Northern blotting. That's for RNA. So Western blotting is for proteins. What was the direction? I'm trying to remember. Wait, it was like here, because you have Northern blotting. Where was it? No, marker. Northern blotting. Southern blotting. Western blotting. And Eastern blotting. DNA. Wait, I think it was like this, right? Yeah. DNA, RNA, protein, after proteins. Beautiful. That's how you remember? Northern blotting is for RNA. RNA here. So now let's start with Northern blotting. Northern blotting. First step is you have your sample. You do RNA extraction. And now you have your, guess what? Your RNA. This is RNA. I'm gonna make it red. After that, you separate it. Ooh, it's still electrophoresis. See, you get your sample, you do electrophoresis. This is your flow. RNA is separated based on size. After that, you have Northern blotting, which is you transfer to a membrane, gel membrane, and that will allow the RNA to be immobilized on the membrane. After that, you put labeled probes specific to the RNA of interest. The probes will bind to the complementary RNA and you have detection happening. Then you just visualize it via X-ray film to produce a signal on the film. Yes, beautiful. Okay, so it's not a debate, but now the question is RT-PCR versus PCR. We're gonna try and do that one. Here. So line in the middle. That line is so straight. One moment. Line. Let's write it. Let's try to make it Much better compared to the one earlier. Okay, so you have RT-PCR and PCR. RT-PCR is reverse transcription. RT-PCR. They share similar amplification processes, but they differ in the starting material and initial steps. So in RT-PCR, you start with RNA extraction. Okay, you start with RNA extraction. This is your RNA, and then let's draw it out. A poly tail. Then you have your primer. Oops, ugly color. Can't be seen. Orange, please. There you go. This is your primer. RNA is reverse transcripted into cDNA. DNA. So how will that look like? You get your cDNA. Now it will be over here. No. I'll just draw it out again so that the steps are clear. So you have the RNA, and then you have your primer. Oh, that's still red. No. And then you have the cDNA over here. Perfect. After you make it using complementary DNA, the step produces it, and that is then amplified, okay, through the PCR process. It just kage bunshin jutsu, this one. Times two. Okay, that is amplification. The steps would be denature, anneal, extension, etc. Nice. So see, we have Power World and we have Naruto now. Oh, anime is so awesome. Next, PCR. You start with DNA as a template. And then next is denature, anneal, and then extend. Denature, anneal, and extend. Denature is just you denature the double-stranded DNA at 95 degrees Celsius, and then you anneal the primer. I think like that's like split like them up around 50 degrees Celsius, and then you extend it. Denaturation into this one. You anneal by adding the primers P1, P2, and you extend it. You have the direction going here and then the direction going there. Nice. See, in summary, RT-PCR will amplify cDNA from the RNA, while standard PCR would just amplify the DNA directly. So that's their main difference. Again, RT-PCR versus PCR, they are the same in terms of amplification, but they differ in terms of starting material and the initial steps. Why? Because RT-PCR is you start with RNA, and then you make cDNA from that one, and then you do the normal PCR steps. But then with PCR, you already start with the DNA. So you just do the denaturation, annealing, and extension directly. Beautiful. Then you have a microchip array. That one involves a lot of different steps to examine the gene expression. Okay, how do we do that? Well, you construct it with DNA clones, followed by, I'll just write it. Don't worry. Here. So microchip array, microchip analysis. You construct the microarrays with DNA clones, and then you do PCR amplification. After the clones are happening, the Kagibunshins, the Bunshins, shadow clones, you are then robotic printed onto the microarray chip. And then for RNA-seq, you have different steps for that one. RNA-seq. So first you isolate the total RNA from the sample. Then you make cDNA and then the next step is just to sequence the cDNAs to provide a detailed map of the transcriptome. So yeah, if you think about it, potential experimental results would be like, does your amount of protein change in your experimental system? And if both the amount of protein and the mRNA content changes in parallel, did it alter because of like a transcriptional mechanism? So you just have to think about that properly. And take note that transcription regulation involves controlling the number of RNA polymerase II molecules that interact with the gene. And when fewer RNA polymerase II molecules are present, the gene is transcribed at a lower level. So, and conversely, if you have a lot of it, then the gene could be transcribed at a higher level. So for example, you have gene X and you have gene X also, but if you have only one RNA, it can lead to one RNA pol. You have one RNA pol 2, and then you have 10 RNA pol 2. So of course you get a lot of more proteins from here, from the one which has more RNA pols. So we could do a run-on assay, but no, I'm fine with not doing that one. It's just transcription. In a run-on assay, you have a gene transcription rate of a specific gene, like gene A, and you measure it by the synthesis of your RNA. First is you isolate the DNA template containing gene A and then RNA pol 2. Despite I don't understand much, your current video is the best video I saw today. That's saying a lot. Oh, thank you for saying that. I try my best. Oh, by the way, your music video is coming out this month and next month with music. Yes. And so sometimes I do a mix. I do this one, science, and then I play Pal World. I play Pokémon also, and a lot of other things. It just depends on the weather, and the timing. And this, okay, that's fine. You have different genes. It's okay. Oh, you have a low potassium regulation of NAK-ATPase subunit gene expression. This one, you just need to check. Does the findings suggest that low potassium induces an increase in sodium potassium ATPase subunit and mRNA abundance? So if you want to explore that, just check a portion of the 5' end region and maybe you'll see something. Alright. And then you could do an in vivo response in vivo approach to response element definition, and that just means create the transgenic mice that carry the specific changes in the DNA. Alright, great job. And then if we want to check transcription factors, this is where it gets interesting, I think, because you can check more here. Wait, marker. Transcription factors. yeah. So for transcription factors, there's a lot, right? But if we use something like the electrophoretic mobility shift assay, these sound like Harry Potter spells. And then you have the chromatin immunoprecipitation assay, like CHIP. That will help you study the protein DNA iso interactions. Because, for example, chromatin immunoprecipitation assay. That one is protein and DNA interaction. Because in CHIP, the DNA is cross-linked to proteins. You're linked forever. The red string of fate, Mitsuha and Taki. That is Mitsuha, this is Taki. They interact. I mean, they're cross-linked. And then the chromatin is fragmented. And then an antibody specific to the transcription factor will pull down the protein, which is, take note, attached to the DNA. After you pull it down, then you identify then you isolate the DNA that can be analyzed to identify the regions that the transcription factor binds to in vivo. Nice. And then you have EMSA, which is this the gel shift assay to study protein DNA interactions. Also starts by labeling DNA with cell extract and etc. I think is that. Oh, CHIP, we already discussed CHIP. You lyse, break it, you see the amplification. You can amplify it also by PCR. Okay, so mini question: how is protein quantified using Western blot? They're separated by size using SDS page. They're transferred to the membrane and they're probed with a primary antibody specific to a target protein. And a secondary antibody is conjugated with HRP to amplify the signals. Northern blotting, how does it quantify mRNA? You have extract the RNA, you do electrophoresis, transfer to membrane, add the probes, and do X-ray. Oh my goodness. I think that is all for transgenic mice and genetic engineering and experimental design in transcription. I will now take a break, but thank you so much for coming to this mini stream. And was it a short stream? I'm not sure. But yeah, thank you for watching. That is all for today. Cheers. Yes, this one. Bye bye.